Hello everyone. Good evening and a warm welcome to you to this webinar. The topic for today's webinar is testing ASXL1 gene variants in malignant myeloid diseases with case studies. At the outset, I would like to thank Dr. Rahul Bhargava for organizing and coordinating this webinar. Our speaker for today is Dr. Arun Kumar. Dr. Arun holds a PhD in genomics and is a scientific affairs manager at MedGenome Labs Limited. Before I hand over to Dr. Arun, I wanted to inform you that if you have any questions or comments, you can type them out in the Q&A window of the Zoom conferencing browser. We will also be sending across the recording of this webinar in case you missed out any points or would like to revisit some portions later. So without any further delay, I would now transfer the host to Dr. Arun, who will take this webinar forward.
Thank you, Hardik. Thank you, Dr. Rahul. I welcome you all for this evening session of I'm Arun Kumar. I'm a geneticist and I work as Scientific Affairs Manager at MedGenome Labs. So today I will be talking on the importance of ASXL1 testing with patients with malignant myeloid diseases. And I'll also cover a few case studies as an example of how this is being useful. The agenda of today's presentation is, we'll start with the basics of ASXL1 gene, the background and biology. Then we will try to understand the role of ASXL1 in hematopoiesis and the relevance of ASXL1 mutations in various myeloid malignancies. And what is the current hypothesis of developing AML in patients who have the ASXL1 mutations. And finally, we will do a few case studies to understand how this is seen clinically. So to give a brief background of the myeloid malignancies, as we know, myeloid malignancies are characterized by aberrant clonal expansion and differentiation defects, particularly of the hematopoietic stem cells and the hematopoietic stem progenitor cells or the myeloid progenitor cells. Now, these myeloid malignancies are particularly, we are talking about the myeloid lineage of hematopoiesis and not the lymphoid. And most of the myeloid malignancies, as we all know, is associated with high mortality and also morbidity in a few cases, particularly because of the few limitations in the availability of therapeutic agents, and also the relapse rate is very high in myeloid malignancies. So various research groups across the world are trying to understand the genetics of these myeloid malignancies, particularly with respect to the mutational landscape of these myeloid malignancies and the genes associated with it, to identify various genetic factors and mutations that can be of prognostic and therapeutic value in the future. One of the widely mutated, commonly mutated gene in various myeloid malignancies is the ASXL1 gene. And this is one of the genes that is now widely investigated by researchers across the globe. So this talk will particularly focus on this ASXL1 gene. ASXL1 stands for additional sex chromosomes like 1. So this gene was first identified in a Drosophila model, the fruit fly. So in the Drosophila, it was identified as an additional sex chromosome gene, which is, and the human ASXL1 is a paralog of the Drosophila's ASX gene. So this Drosophila's ASX gene was originally identified as an enhancer element. So enhancer is a group of proteins that aid in the transcriptional activity of genes. And this ASX in the Drosophila was originally identified as an enhancer of trithorax and polycomb group of genes that regulate the Hox gene expression. So this trithorax and polycomb genes are evolutionarily very conserved chromosome or chromatin modifying factors. We'll just visit in a while what these are. So basically these interact with the ASX, and the ASX acts as a control for the expression of Hox gene. So this is the brief history of how ASX was identified. In the humans, the AXS has been identified to be primarily involved in chromatin remodeling. So the expression of Hox genes, the Hox genes are very important in organ development during embryogenesis, and that was controlled by ASXL1 protein. And recent evidences also show that ASXL1 protein is involved in promoter methylation of certain genes. So just to give a brief background of what chromatin remodeling is, let us visit the basics of chromosomal arrangement or the human genome organization. So the human genome is a very large genome that is compactly packed inside the cell's nucleus using an assembly of proteins called as the histones. So these histone molecules, you can see here labeled as H3, H4, etc., form to make complex, and DNA gets wound around these histone molecules, and this particle is called as a nucleosome particle. So the DNA is tightly wound around these histone particles, and it is organized compactly inside the nucleus. But suppose there is a gene which needs to be expressed for a particular function of a cell. And that gene, say in this cartoon, it is shown as blue in color, is wound around the histone particle. The expression of the gene or the RNA synthesis cannot take place because the RNA or the other regulatory proteins cannot access the gene wound around the histone particle. So a process called as chromatin remodeling complex, it's a group of protein, does a process called as chromatin remodeling, where it changes the certain residues, amino acid residues of the histone particle, and loosens up this particular part of the gene free, so that the regulatory elements or the enzymes can act upon this particular gene, and the gene expression and other activities take place. So this set of proteins, chromatin remodeling complex, a very complex group of proteins involving more than 40 to 50 different types of proteins. And it is suggested that our ASXL1 is also involved in this group of proteins, and it is involved in this process of chromatin remodeling. So in a nutshell, ASXL1 is involved in the epigenetic control of gene expression. So moving forward, the ASXL gene is located in the chromosome 20 of the human genome in the Q arm. And this gene has 13 different exons. Initially 12 were estimated, and with the recent build of the human genome, the latest build HG19, it has been identified that it has 13 exons. And the 13th exon is the largest or the longest exon. And this protein has 1,541 amino acids, and the protein has various domains in it. So the N-terminal domain is called as the PHD domain or the plant homeo domain. This is the most important domain which is involved in the activity of this ASXL1 protein. So this PHD domain is the one that actually recognizes the other proteins that are involved in the chromatin remodeling complex and other transcriptional factors and enhancers that are involved in the expression of gene. So that is the most active and well characterized region of this ASXL1 protein. So some of the molecules that this PHD domain interact with are called as PRC1, PRC2, etc. The gene on its C terminus has ASXN and ASXM region. So ASXN stands for conserved N-terminal. Sorry, this is the C terminus, this is the N terminus. Conserved N-terminal region, and this is called as the conserved domain in the middle part. There is also a domain called as NR box, which is there in which is called as a nuclear box. Now, please note that we still have not understood and evaluated the exact molecular nature of this ASXL protein. What are all the other proteins it is interacting with? What are all the exact molecular pathways that it is involved in? What is the exact function of this protein, etc., is still being investigated. Now, various mutations in the exon 12, particularly somatic mutations in the exon 12, have been attributed to different myeloid malignancies. Particularly, most of these mutations lead to a frame shift in the reading frame. So when a mutation causes a frame shift, the whole protein becomes nonsense. And many times these proteins, these mutations lead to an early termination of the protein synthesis, so that this PHD domain is not produced in the protein at all. So these are some of the commonly seen mutations, of which this particular 646 mutation, glycine 646, which becomes a tryptophan, is the commonest mutation, and that accounts for almost 50% of all known ASXL1 mutations. So, and this is associated with most of the myeloid neoplasms. And it has been suggested that given these evidences, these mutations actually are loss of function mutations. Although there are a few debating concepts going on around it, but right now it is understood that they are loss of function mutations. Moving further, this concept of myeloid malignancies and ASXL1 was identified because ASXL1 was frequently mutated in many different myeloid malignancies. To name a few, CMML patients showed almost 40 to 50% of them had ASXL1 mutations. AML patients with myelodysplasia related changes, 37% of them had ASXL1 mutation. Likewise, patients with myelofibrosis, 32%, MDS patient ranged from anywhere 15 to 20%, acute myeloid leukemia, 5 to 15%, and various MPNs such as PV and ET, around 5%, 7 and 4% respectively. So given that ASXL1 mutations are seen in so many different myeloid malignancies and not lymphoid malignancies, people started to research more about this ASXL1 mutations, the gene, and its role in the hematopoiesis. So, to understand the role of this ASXL1 in the normal hematopoiesis, the researchers first started doing experiments in mice models. So, they developed mice models with deletion forms of ASXL1. So you can artificially induce a deletion in the mice of the particular gene by translocation experiments, and they tried to see what were the effects of this deletion. So first thing what they identified was that mice which had deletion of ASXL1 gene had an impaired myeloid differentiation. And secondly, heterozygous deletion, that is in one chromosome, if the mouse has a healthy ASXL1, in the other chromosome, if it is deleted, those mice developed MDS or MP. And ASXL1 loss also exhibited, the mice with ASXL1 loss also exhibited hematopoietic stem cell reduction, hematopoietic stem progenitor cell reduction, and most importantly, the regeneration capacity or repopulation ability of the hematopoietic stem cells were impaired. Lastly, it was also found that ASXL1 deletion forms, deletion in the mice, cooperated with NF1 genes haploinsufficiency, and it activated multiple oncogenic pathways such as NRAS, MYC, etc., and it promoted myeloid transformation. So having done these experiments in mice, researchers also wanted to see the effect of this ASXL1 in human cells. So a variety of studies took place. I'll just mention a few highlighting ones. So in the human cells, bone marrow stromal cells which were derived from CMML patients were taken, and it was found that they had a decreased expression of ASXL1. And the cord blood cell with CD34 positive. CD34 is a stem cell marker, and it was found that when you knock ASXL1 gene in the CD34 plus cord blood cells, it resulted in a reduced erythropoiesis and impaired erythrocyte enucleation. Again, please note that this is a myeloid lineage. Silencing of ASXL1 by lentiviral transduction again. In CD34 positive bone marrow cells, it altered the granulomonocytic differentiation, and increased number of multipotent mixed lineage colony-forming units were found. Similarly, colony-forming units of granulocyte macrophage were also high, and the granulocyte colony-forming units were also high. Thus, these studies, both at animal experiment level and at a human cellular level, where it demonstrated that ASXL1 was very important in maintaining the normal hematopoiesis of an individual. And these studies also showed that deletion of ASXL1 or impairment of the ASXL1 expression led to the promotion of myeloid malignancies in those subjects. Next, moving further, let us try to understand the role of ASXL1 mutations in various myeloid malignancies. To start with, we'll see the case of CMML. So it has already been shown that almost 40 to 50% of CMML patients had ASXL1 mutation. And it is a marker of poor prognosis. And ASXL1 mutations frequently coexisted with TER2 mutations in the CMML patients. TER2 is one of the markers which is genes that is commonly mutated in CMML patients. But upon subset analysis of TER2 patients, they found that patients with both ASXL and TER2 mutated patients had a shorter survival. Whereas TER2 mutated alone with ASXL wild type were categorized as a favorable risk group in that particular study. Additionally, interestingly, they also found that the patients with ASXL1 mutation had a poor response to hypomethylating agent therapy in one of the studies. So the overall response rate with ASXL1 mutated patients was lesser. As you can see here, on the x-axis, you have the overall response rate, and on the y-axis, you have the genes that are mutated. So the ASXL1 mutated group is towards the left side, meaning that they have a lower overall response rate. On the other hand, those with TER2 mutation or NRAS mutation had slightly a higher overall response rate. Interestingly, among this TER2 mutated group, if when they removed off those with a coexistent ASXL1 mutation, the TER2 mutated and ASXL1 wild type group had a much higher overall response rate. So it also, ASXL1 mutation also shows that the response to certain therapies can be impaired. Moving further, in AML, ASXL1 mutations are found in almost 15%, 5 to 15% of the cases in various studies. And it independently confers a poor prognosis. So the point here, independently, is important because independently, it does not act in liaison with any other genes to cause the poor prognosis. Presence of ASXL1 itself is a poor prognostic indication. Then a study of 740 AML patients in 2013, a very seminal study, they tried to identify various factors in AML patients with and without ASXL1 mutation. And they found that of these 740 AML patients, 17.2% were ASXL1 mutated. Now, interestingly, in this cohort, they found that males had a higher frequency of ASXL1 mutation than the females. And this has also been replicated in few other cohorts. So almost 23.5% of the males had the ASXL1 mutation, whereas 9.9% of the females had ASXL1 mutation. And the median age of the patients with ASXL1 mutation was higher than those without the ASXL1 mutation. For example, here 71.8 is the median age of patients with ASXL1, and 61.8 was the median age of those AML patients without ASXL1. And it was present in 35% of the patients of AML who had a karyotype abnormality. And we also know that AML is AML patients will carry a variety of other gene mutations, such as FLT3, NPM1, RUNX1, DNMT3A, etc. Surprisingly, they found most of frequent combination mutation of ASXL1 in AML was with RUNX than with other mutations. Not that these are impossible, but for some reason, surprisingly, people with RUNX1 mutation also had ASXL1 mutation. As a FYI, recently a group has started to study the expression of RUNX with, which is to see whether it is controlled by ASXL1. The research is on the way. And in this particular cohort, again, they found that coexistence of ASXL1 and RUNX mutation was a poor prognosis in the AML patients. They did an overall survival analysis of AML patients, and they found that ASXL1 clearly conferred inferior overall survival. For example, you can see here in the Kaplan-Meier plot, This is the, on the x-axis you have the months and the y-axis is the percentage of people with the overall survival. And the median overall survival in the wild type, that is the healthy those with healthy ASXL1 gene, was 62.2 months. Whereas in the ASXL1 mutant people, it was only 11 months, almost 5.5 times lesser than the wild type. So it clearly shows how bad the prognosis is for people with ASXL1 mutation. When they stratified the same as less than 60 and more than 60, again, they found that the ASXL1 mutation conferred a lower prognosis and the overall survival rate was much lesser. So here, in this case, you can see that 2-year survival was 60% and here it was 78%, much higher in the less group. Whereas those with more than 60 years, ASXL1 median overall survival time was 36.3% and 11 only for the mutant group. And you can see that this difference, particularly the first overall group, it is highly significant. P-value is pretty significant. So the take-home message is very strong evidences suggest that ASXL1 confers inferior overall survival in AML patients. Interestingly, in the same study, they tried to categorize the AML into the etiology of the AML. So patients who had a de novo AML, or it's a primary AML as you can say, or therapy-related AML, they had a much lower frequency of ASXL1 compared to those with secondary AML. It's almost more than twice the frequency. So those with secondary AML, or in other words, those patients who had an earlier dysplastic bone marrow dysplasia or MDS patients who developed AML later, those secondary AML cases had a higher, significantly higher frequency of ASXL1 mutation. This is again, I would like to, I wish you to pay note. And this was also highly significant statistically. So ASXL1 mutations were significantly higher in secondary AML cases. Moving further to the frequency of ASXL1 mutation in MDS, or the role of ASXL1 mutations in MDS, the frequency ranges anywhere from 15 to 20%. And in a study in 2014 of 362 MDS patients, they found 17.1 had ASXL1 mutations. And the mutations were associated with a variety of other genetic alterations, such as trisomy 8, mutations in RUNX, EZH2, IDH, NRAS, etc. And but interestingly, SF3B1 mutation was independent of ASXL1 mutation. Again, it is only an observation. We really do not know why it is so. So those who had SF3B1 mutation did not have the ASXL1 mutation. So the molecular nature of this observation is yet to be elucidated. And ASXL1 mutation was again an independent poor prognostic factor for the survival of MDS patients. As you see here again in the plot, the x-axis you have this overall survival in month, and the y-axis is the proportion of people who are surviving. So of 300 ASXL1 wild type, that is the healthy ASXL1 gene carriers with MDS, the median survival time was 69.9 months. Whereas in the mutated people, it was only 21 months. So you can see the difference in the prognosis of ASXL1 and the healthy people, and the ASXL1 wild type people. And in a study in 2010, interestingly, they identified another factor. So this was a study with around 250 MDS cases and a few AML cases. And on the x-axis, you have the frequency of ASXL1 mutation. On y-axis is the various categories of MDS. So those MDS with 5q deletion or refractory anemia had a frequency of around 6% and 7% respectively. And refractory cytopenia with multilineage dysplasia had no ASXL1 mutation at all in this particular cohort. Refractory anemia with ringed sideroblast, or almost 29 patients, all of them had, the frequency of the mutation was around 17%. While the refractory anemia with excess blasts class 1 and class 2 had a high ASXL1 mutation of almost 31%. Likewise, AML patients who were, it's a secondary AML post MDS, they also had a pretty high frequency of ASXL1 mutation of up to 24%. Now, the point here is that these class of patients, those with excess blasts and the AML post MDS are late stage MDS patients, whereas these people here are early stage MDS patients, and thus ASXL1 mutations are more frequently observed in late stage MDS patients than in early stage patients. So, current hypothesis of AML development from hematopoietic stem cell is this. With respect to ASXL1 mutations and other mutations is this. If there is a hematopoietic stem cell, there could be a first attack by TER2 mutation or any other genetic alteration that can lead to the progression of AML. You do have other mutations, genes such as NPM1, FLT3, IDH, etc., which can mutate independently or in liaise with other genes and develop AML. So this is the case of primary AML. Whereas if the bone marrow is dysplastic, or if it already has myelodysplastic syndrome or primary myelofibrosis, as you see in MPN cases, because of the presence of a initial hit by TET2 or RUNX1 mutation, it progresses further to get ASXL1 mutation. This trigger of ASXL1, a mutation by ASXL1, triggers the patient to progress to a pre-leukemic state and leading to secondary AML. Of course, this constitutes almost 1/3 of the secondary AML cases. You do have other possibilities of mutations such as CBL or IDH that can also give you the same secondary AML cases. But the point here is that presence of ASXL1 is almost very sure to say that the prognosis is poor and the chance of developing leukemia is extremely high. So with this background of ASXL1 in various leukemias, let us try to see a few case studies. So this is a case of 31-year-old male who is diagnosed with AML by immune phenotyping, and he was tested in the AML risk stratification panel. And we identified mutations in the IDH1, ASXL1, and DNMT3A. So this is the mutational profile, IDH1, ASXL, and the DNMT3A. And these two are classified as alterations with strong clinical prognostic and therapeutic significance. Because IDH1 has a drug target available. Now, ASXL1 mutation, I would like you to pay note to this region where it is a exon 13 mutation, and this is the classical 646th position mutation that we just saw, which constitutes almost 50% of all ASXL1 mutations. And the read depth was 1053. So just to mention this about this read depth determines how confident are we in calling this particular variant. So it is the number of times a particular nucleotide is sequenced in that particular experiment. So I can say here that this insertion G mutation in this particular sample was sequenced 1053 times in different samples. So as a thumb rule for somatic testing, we go for anything over and above 250x depth. For a germline, it is around 180 to 100x. For a somatic, where the mutation load varies, we go for 250x and above. So in this particular case, this ASXL1 mutation was identified in 1053 different fragments of the NGS experiment, meaning that the depth is 1053. And the mutant allele percentage or the mutation load was 14.3%. And as we have already shown, it has a presence of ASXL1, signifies a shorter overall survival in AML patients. So then we go on to describe the various attributes of the ASXL1 gene and its protein. And so it's a transcriptional factor, etc. And the mutation that we identified is a 24 base pair frameshift deletion because of the insertion of one particular G in the cDNA, or in the chromosome 20 in this position between this 441 and 442, a G is inserted. That leads to a frame shift, and that causes a truncation in the protein, 12 amino acids downstream of the 646th position of the protein. So meaning that if you remember that picture of the ASXL1 protein, the whole PHT domain is not synthesized in this individual at all. So that means that the protein is not effective, and we have classified this as a loss of function variant. The minor allele frequency in the exact database was 0.16, and in the healthy 1000 genome database, this minor allele frequency or this particular mutation was not observed at all. And then we go on to describe various studies which correlate or which connect the ASXL1 mutations with AML cases. Now, sir, in the next case, it is a case of a 45-year-old female, confirmed case of AML. And when we tested them, interestingly, we found only the ASXL1 mutation and no other mutation at all. This was a very interesting one because you generally tend to identify other mutations in AML, but this was only ASXL1 mutation. And again, this was there in the exon 13, but in the same 646th position, where 12 amino acids are, the termination takes place, 12 amino acids goes to 646. At the cDNA level, it is a 1943 position, there is a duplication. And the mutant allele load was 15.2%, and the sequencing depth was 983x. This is a young patient's case, might not be juvenile, closer to juvenile and young adult patient, 13-year-old male, a case of AML. And we identified mutations in KIT as well as ASXL1 genes in this subject. So these are the two KIT mutations that we identified. One is at a frequency of mutation load of 2.2%, and this is at a mutation load of 29.8%. Whereas the ASXL1 was, again, the mutation was there in exon 13, and the mutation frequency or the mutation allele load was 17%, and the sequencing depth was 915x. Now, this, when we dug up the literature to understand this, we found that in one study of 204 pediatric de novo AML cases, not secondary, de novo AML cases, ASXL1 was not associated with poor outcome. So this particular study again points out that it is imperative for us to understand, know the age of the patient to understand the prognosis of ASXL. So one study on juvenile or pediatric cases of de novo AML, it showed that ASXL did not have a poor prognosis as it shows in elderly people. Moving further, this is a case of 70-year-old male. He had a secondary AML. And we identified a variety of mutation in this individual in various genes such as RUNX, TET1, ASXL, SRSF2, and STAG2. So this is the RUNX mutation, this is the TET2 mutation, and ASXL mutation was also seen again in the exon 13 of 24.6% mutant allele load. So here is a case where again you can see RUNX mutation is there along with ASXL1 mutation, and this could be a classical case where in that diagram I showed you, from a hematopoietic stem cell, it progressed towards secondary AML. So this could have been the trigger point, and moving further, the patient could have accumulated various mutations, including ASXL, and developed secondary AML. And we also identified mutations in SRSF2, NRAS, STAG2 at various mutation allele depths. And we also identified two variants of uncertain significance in the genes of SETB1, SETBP1. Two different mutations were identified. So you can see that a patient with secondary AML can have such a different repertoire or such a different array of mutations in so many different genes. So this patient has mutations in 6-7 different genes, and in one gene he has 2 different mutations. Of course, these two were variant of uncertain significance. Of course, the prognosis is going to be poor in this patient just because he has ASXL1 mutation. This is a case of 48-year-old male suspected with MDS, and we identified a single ASXL1 mutation in the exon 13, and it was 34%, and the depth was 1101x. And again, this is, now you can see here, although it is in the exon 13, it is not that classical 646th amino acid position. It is there in a different amino acid position, which is 859. And the mutation in the cDNA is also there in the 2577 position, where there is a single deletion that has taken place. Again, this mutation is also leading to termination and impaired production of the protein. And again, in the case of MDS, 15.8% of MDS cases are known to contain ASXL1 mutation. And in a study of 170 patients, they have shown that the transformation to AML was much higher in patients having ASXL1 mutations. This is the last case where a 56-year-old female was diagnosed with AML. We identified clinically relevant mutations in TET2, NPM1, FLT3, and DNMT3A. So these are the list of genes that we identified, mutations and the genes that we identified. It was having, the patient also had FLT3-ITD positive. But the ASXL1 mutation that we identified was in the cDNA position 890, which corresponds to exon 10. Now, please remember, I said there were 13 exons, of which the 13th exon is the most important one. And all studies until now had been focused only on this 13th exon. Whereas this individual had a mutation at the 297th amino acid, where a threonine became a methionine, which corresponds to the exon 10 of the gene. And it was sequenced at the depth of 1913x, and 47.8% was the mutant allele percentage. Now, this mutation, although the gene is known, this mutation is not understood, so it is classified as VUS. So we show that this mutation has not been characterized sufficiently in cellular studies or biochemical studies, and therefore the impact or the relevance of this mutation, even though the gene is well understood, this particular mutation in exon 10, the relevance of it in cancer is still speculative. That's the reason why we have classified it as variant of uncertain significance. And the functional impact of this variant and the clinical significance of this variant is not at all well documented in the medical literature. So this is a case of, it was a recent case. It will be interesting to see, or at least understand, what happens to this patient, whether she is progressing well or poorer, to see what is the effect of this mutation. Of course, you need cellular studies and functional studies to classify this variant into a pathogenic one. So the key takeaways from today's webinar are: ASXL1 protein controls the expression of genes by chromatin remodeling, or in other words, epigenetic control. Most of the somatic mutations in the ASXL1 gene are in the exon 13, particularly in the PHD or the PHD domain, which leads to a loss of function. A duplication of G in the position cDNA position 1934 causes a change in the amino acid at the 646th position of the protein, and it leads to a frame shift and accounts for almost 50% of all ASXL1 mutations in myeloid neoplasms. And ASXL1 mutations commonly observed in CMML, and it is again very common in late stage MDS and secondary AML cases. ASXL1 mutations independently confer poor prognosis of the disease. Thus, looking at all these evidences, and even the guidelines suggest various guidelines, it is suggested that we should include ASXL1 gene in various myeloid malignancy testing, so, or the genetic testing of various myeloid malignancies. With this, I thank you all for this opportunity. I hope you understood what was this, and I'll be very happy to answer any questions. Thank you.
Thank you, Dr. Arun, for that crisp and insightful talk. I see we have a few questions in the chat and question. We can address them one by one. And I also invite questions and comments from the audience if they have reserved their questions till the end. So
Dr. Arun,
yes,
it was a
doctor.
wonderful presentation.
Thank you, sir.
So, do we do in MPN panel, do you think that ASXL1 along with MPN in JAK2, CALR and MPL, we should add ASXL1?
If you're prognosticating MPN patients, yes, doctor. But if the thing is, diagnosis of MPN as per guidelines today is JAK2, CALR, and MPL. But that alone might not be sufficient for you to prognosticate the patient, for which definitely ASXL1 has to be included.
Perfect.
Because patients with ASXL1 will develop fibrosis or dysplasia in the future.
Okay.
Yeah. So I will answer a few questions here. I will read out the question. Dr. Prasad has messaged. If ASXL1 or any other mutation is detected, the read depth is less than 250x, how is it reported? How should it be interpreted for clinical decision making? A very good point. So if the read depth is less than 250x, in the first place, we don't report it. We redo the assay if it is because of certain technical issues. So if it is less than 250x, the report should not carry a variant at all. If it is only more than 250x, do we report it? And then only we will be confident. The logic is that, say in one case, we saw the ASXL1 mutation was there in 15% mutation load, meaning that out of 100 cells, only 15 cells had this. Or in other words, when I talk about mutation percentage, if I have a read depth of 100, only 15 reads will have this mutation, will show up this mutation. So a read depth of 1000, when I go, I'll have 150 of them showing the mutation. So more the read depth, more confident am I in assigning low frequency mutations. I hope that answers your question. Second question, have you encountered a case of MDS which was ASXL1 negative initially, then turned on progression to second AML? I'm sorry, we have not encountered such a case because personally, we have not come across such a case. Yeah. Which requires 2 times MDS prognostication testing. And to my knowledge, we have not come up with such a.
So, Dr. Arun, before we proceed ahead, for the clinical group, can you tell the US best practices that with every time when there's a patient progresses, does any center in the US does NGS repeatedly?
Dr. Rahul, I can answer this. I'm Ram here.
Hi, Ram.
Yeah. So the same query I asked our CAP auditor, who was basically an oncologist, more of a solid tumor person. I've asked them whether in the MRD context, I asked them whether in US, at least for some of the critical and very well established prognostication markers and mutations, whether you repeatedly do an NGS. So her answer was, as per the consensus, there is no guideline, specific guideline, but as per the consensus among the groups there, some of the markers they do, if not multiple times, and more than once they do. But it is not a routine practice since the data is still coming out, especially in the context of MRD. Few markers are now well established, but some of the markers, yes, more than once they do.
Thank you, Dr. Ram. So what is the testing method, NGS versus real-time PCR? There is one question. So to answer that, we follow only NGS because we don't do only the hotspot sequencing. We sequence the entire coding region of the gene. And the results for that is useful because I just showed you various reports where the mutation was not in the same locus alone. So the mutations were there in different loci of the gene. Whereas when you do by real-time PCR, you will target only that particular mutation alone, so that the ability for you to, or the chances for you to identify novel mutations is lost. So that's the reason why, at least for these cases, NGS is always advisable. And next one is, case 1 had an insertion and it was described as a deletion in the description. Okay. So let me just go back to that. No, it is given as a insertion in the description also. So I don't think we have given it as a deletion. And is allele burden significant? So allele burden significance is a very relative term again. So generally you can have it as an idea that higher the allele burden, more is the aggressiveness. But unless we have a specific guidelines to say what is the significant allele burden for this particular gene, we cannot really comment on that. There are no guidelines saying that, but it is more empirical in nature. And have you encountered ASXL1 with NPM1 and biallelic CEBPA mutation in de novo AML? If yes, how does it impact therapeutic decision making? Sorry, we have not encountered such a case till now, to my knowledge again. What should be the plan for adults with ASXL mutation and myeloid malignancy? Should transplant be considered in all post-remission? So this is a question that I think Dr. Rahul Bhargav would be better to answer. But as a molecular biologist or a geneticist, to me, what I understand is that ASXL1 mutation will definitely lead to, or the people with ASXL1 mutation tend to have leukemic malignancies in the future. So if that is the case, if they are at high risk group, what is the clinical decision is something that the clinician should answer.
So I'll just answer that.
Yes.
So when we talk about ASXL1 as a poor prognostic marker, nobody knows whether doing a transplant is going to overcome that or not. One thing, so it is a poor prognostic marker. It entails that you should do something more. But what more? Does transplant overcome ASXL1? Nobody knows an answer for that. More and more people in the world of myelofibrosis, they are saying, even if the patient is intermediate 1 and 2, intermediate 1 where there's a transplant is not indicated, but you have an ASXL1, that becomes a soft target or soft indication to do early transplant. When it is a CABPA, biallelic and ASXL1, it will confer poor prognostic marker. In our case, if there is a matched sibling, we will like to proceed with a bone marrow transplant. But for an haplo and match unrelated donor transplant, I'll be skeptical. So, Arun,
yes, doctor.
The question, what I'm trying to ask is how significant, when you say mutant allelic frequency, at what level we should consider that as significant? What is the frequency at which we should consider that ASXL1 will become a poor prognostic marker? So you have shown a 323 patient data.
Yes.
What level? Any level?
Any level. It was any level. So there was no, exactly, doctor. So there was no subcategorization based on the allele burden.
Okay. So should I read the questions then?
Yeah, I'm just doing that. One is T821 in a 50-year-old male, CR1 after 3+7. NGS showed 0.6% allele frequency, read depth of 950x ASXL1 mutation. Should he be transplanted? No MDS, no mutant burden. So I don't know if it's a hypothetical question.
So Sumit, I would say either way around 821 patients are not doing well. If your center is well equipped in doing and reducing the mortality by less than 20% and can give a 50 to 60% survival, then you should proceed ahead and do a haplo transplant. Otherwise, you can just wait till the time patient relapses. Either way around, if at 950 depth, this ASXL1 is positive, sooner this clone is going to grow up and will create a problem. So 821, we are all defining, but criteria-wise and guideline-wise, except the C-kit, nobody is talking about ASXL1 and jumping on the haplo transplants in 821. We have seen patients a few years back where they were positive, one patient who ultimately relapsed. Either way around, 821 for us is not doing so well. If MRD is positive, C-kit is positive at any moment of time, at 3 months after induction, after consolidation, we tend to take them up for the transplant.
Thank you, doctor. The next question is, apart from NGS, what other methods can we use for detection of ASXL1 mutation? And is there any disadvantage of other methods? So to answer this, you do have variety of other methods, but NGS is the preferred method. The reason is that NGS has a higher sensitivity to detect even low frequent or low allele burden variants. And two, NGS detects even novel variants. Whereas when you do by a real-time PCR, you don't identify novel variants. You can do Sanger, but the disadvantage of Sanger sequencing again is that it requires more than 20% allele burden to identify a variant. So NGS is the best method as presently available especially for rarer mutations like ASXL or other genes, where you can also do multiplex and where you sequence 20, 30 genes together so that it all comes in a single report. So NGS is the best method that is available presently to identify these mutations.
Arun, one question is any significance of high VAF of STAG and last two mutation in case 4? One of them was 80%. Was it germline?
Yeah, that's a good question. So I'll just go back to that.
It's asked by Richa.
Yeah. 1 second. Yes. So this, in all probability, is a germline variant, which is there in the variant of uncertain significant. But again, we have to test it using buccal swab or other source to see if it is germline. Whereas the ASXL1 was only 24%. I don't think any of these are germline in nature. Anything more than 50, around 50, is a good point. And this tag is again 80.2, could be germline. I have to look into this particular mutation and the details of this mutation before commenting further. But yes, just looking at the allele frequency or the mutation allele burden, it's very high, could be germline. Again, we can test it with the Sanger sequencing to see in a
Arun,
buccal swab. Yes, doctor.
So is there any significance of low and high allele burden? Is AXL1 mutation present in normal cell as wild mutation? And then a comment, excellent presentation, Dr. Arun.
Thank you. Thank you, sir. So there is a disease which I did not touch upon detail. There is an inherited disease called as Boring-Optis syndrome, which is caused by germline mutations in the ASXL1 gene. So it is an autosomal dominant way of inheritance. So certain individuals can have germline variants in ASXL1 and they can develop this particular disease. But all the mutations that I talked about now, especially in the exon 13 mutations, are all somatic in nature. And most of the ASXL1 somatic mutations are there only in the myeloid malignancies. I hope that answers the question.
So Shirin Gupta is asking, what is 24 base pair deletion in case 1? Is it a frame shift or a deletion?
This is the case 1. No, it's a 24 base pair frame shift deletion in this chromosome. So because of this insertion, there is a frame shift that has taken place, leading to a deletion in the protein.
Oh, okay. So insertion leading to a deletion of the protein. That's what you're trying to say.
Yes, yes, yes.
Okay. So do you mean AXL1 mutation in young patient is not bad as compared to elderly?
Again, with the available literature, yes, because there were a few studies in young patients with ASXL1 mutation. They didn't have that bad a prognosis as that of adult patients. But again, please note these studies are still in infancies. And especially, getting a cohort itself is difficult. Young patients with the myeloid malignancy with ASXL1 mutation will be a very small cohort, and the follow-up needs to be done for a longer period of time to come to conclusion. But presently, with the current studies, that is the case. But again, please note, guidelines has not said anything about young patients with ASXL1 mutation.
So is the risk stratification of AML, the ASXL1 mutation refers to only exon 13 mutation or any mutation?
Exon 13 only, specifically exon.
So risk stratification for AML is only exon 13. So Arun, you're highlighting a very nice point that when we are reading either a test report as positive or negative, we need to understand that the genomic classification, outcome based on genomic classification is based on particular mutations. Exon 13, exon 10 or whatever frame shift mutations, FLT3 and so on and so forth. Is that right?
That is absolutely correct. Also, I wouldn't say it's only exon 13. I'll say another thing. Suppose if there is a mutation in exon 10 and that is a deletion in exon 10, it is going to affect the exon 13 coded protein region also, because it's a frame shift again. So we have to also see the effect of the mutation. So that is what we show in the description. So we also predict the effect of the mutation, and we do in silico predictions to show that. So again, here it is in exon 13, but it says frame shift and TER. TER means termination. Whenever you see this terminology of TER in the report, in the particular genomic report, it means that the protein is terminated earlier, and that is a very bad thing for the protein.
Okay.
So yes, majority of the mutations are exon 13, which are very bad, but nonetheless, if at all we identify such terminating mutations in exon 10 or exon 9 or any other exon, which can also affect the exon 13, it can still be bad.
So why we should do more for patient with ASXL mutation, a young patient with ASXL1 and AML and no matched sibling, what should be the plan post-induction? In young patient, as he has shown, we can actually wait, but my hunch is keep on collecting the data. Sooner or later, this is going to become a bigger bad molecule, which is going to have a prognostication effect in the coming years. So just keep on collecting the data. As per the data today, if the ASXL1 is positive in younger patient, there's no criteria to do a transplant. So if Dr. Ram there, Ram, is this a good molecule? Is it a good gene to follow them up for MRD? Because certain genes are labile. Is this a good gene to consider as a MRD marker?
Don't know, sir. So far, not many of the people have been doing. In fact, yesterday I was looking at several literature on ASXL1 and MRD. I couldn't get a good one. Something like a FLT3, people are not doing ASXL1.
Okay. So how often do you encounter slippage strand mispairing in ASXL1 testing?
Arun, you want to take this?
Sorry, what was it, doctor?
How often do you encounter slippage strand mispairing in ASXL1 testing?
So basically, they're talking about allele dropout.
Allele dropout. It will be very limited in our experience, doctor. We never come across that much.
So, sir, you mentioned about loss of function mutation. Any significance with gain of function mutation?
So, till now, we have not identified, or literature has not shown gain of function mutations in ASXL, primarily because, gain of function mutations are generally seen either in enzymes or in cell receptors. And this is a chromatin remodeling complex protein. So we have not identified, or the literature has not shown such evidences. But again, the point is that since the PhD domain is losing, we call it a loss of function.
Okay. So just for a recap, Arun, FLT3 was loss of function, or FLT3 was gain of function?
FLT3 was gain of function.
Okay.
That's why you use a FLT3 inhibitor. Yes.
Okay. FLT3 is a gain of function, where AXL1 is a loss of function.
Exactly.
Okay. So life is becoming tough, huh? You people are making, for sure, we are making loser here more. So Abhay has written excellent presentation, and my ability to interpret report is now better.
Thank you, doctor. Thank you.
So with this, we come to an end for today's session. There's no more questions. 1 hour we have been through and through. Very nice. Next week will be TP53 and 17P.